So after heavy dose, yeah. સવાર heavy થઈ જતું હશે ને. Yes, so now let's move for the next point. That is the microbial growth. How we can measure or assess that anything is changed, microbial growth. Like someone is having fever, someone is checking, right? Is there fever or not? If we do have, some persons may feel by him or herself that I am not feeling good. Other person can let me touch, right? So it is the some direct or indirect. Here, why I'm just giving such example, that measurement for anything, it may be a direct or an indirect way. Like just I'm touching, that someone having fever or not, that is a direct method. I may have the thermometer. There are the several kinds of, you can put something in mouth. That's also nowadays that during the corona conditions, they have the laser light, putting here and there. You have any temperature measure, it's or not. Okay, so it's a direct way also. Indirect way to confirm that infections is there or not. Or we may go for the another method also. Okay, some like we may take the bloods for the going for the analysis. So maybe it is a invasive technique, non-invasive. Do you know what is invasive and non-invasive? It is a medical terminology actually. Invasive method, non-invasive. Invasive means we are going to either puncture or surgically cut some body parts. Sometimes I want to take the bloods. Okay, so it's surgically something, maybe surgical tools I have used. That is an invasive method. Non-invasive means I'm looking to diagnose some of the disease or something, but I'm not going to use or cut by any means. I may have the like a, if there is a stomach problem is there, I can cut, I can check also. Is there any problem? Okay, I can go for the sonography. Both the way it's possible, right? Sometimes some person has got, nowadays there's some lasers and the microscopic things are also coming, that we not go for the that much level of surgical instrumentations also need to use. Yeah. So there are, in every case, medical sector or any sectors, we may have some direct or indirect. મારે બોલાવા જવા પડશે, બધા હાથ પકડીને હવે આવી જાઓ ભાઈ પ્લીઝ. રામથી આમ જોયા રાખવું પડશે કે ભાઈ ક્યાંથી આમ આવે. So there is a way to measure the things in direct confirmations or indirect. So several methods are there. Here we are looking for the microbial growth. Okay, what is the microbial growth? That in given media, either it's a solid or it may be in a liquid, we may go for that. Is it a growth is there or not? It is our main target. What we can do generally when we are keeping the tubes or any plates or any broths in a flask, okay, keeping in the incubator, right? On the next day we are coming, we find either colonies or the broths, you may find that somewhat turbid. So by assessing, yesterday's if you have taken the pics, or you may have kept the two things, one is the control, one is the experimental. In control, you may haven't put anything. It is remain as such idle, another is there. So by naked eye also, you can see. So it is a way to check or measure there is growth is there or not, but otherwise you may go to several methods. First, you may have already learned. That is a 4 plate. That is a direct measurement of microbial growth. We are looking for plate counts. It is the very easiest way for identifications that there is a microbial growth is there or not. You may have some water sample, or you may have some soil sample. Okay, or is the blood or something is taken, you may transfer onto the plate. Maybe you go for the spread plate or a pour plate. Otherwise also sticking also you can do. Any plating methods you may go. So our sample, okay, our sample do have any microbes or not. That is our target. So it's a direct. You may directly inoculate your plates. So it is the method already we have learned. That is what is been done in 44 plate method. Last experimentations we have done, right? So something is about 40 to 5, 450 degrees. That is a warm. Our agar is there. There is a molten agar. You are going to spread some of the way. Yeah. Earlier I have also told. Here in picturizations also people are doing what? They pour first, then they may, like in a spread student also. They put some microbial sample. But in this process, it's mainly been observed that during this time, it gets solidified. I think someone has prepared some plates that is also in a slanted here and there. Because when you have started to pour itself, it is got solidified. Right. So that is a major problem. Another problem is also what? The temperature. Some has claimed that I don't have any sort of the growth into plant. So it may be a hot and you have inoculated. So it is killing. It is something like when you are going, I think given the example of the tea. When you are going to in a canteen, cup is near to it, even you will just stick, whether it is hot or not, otherwise we blow the air. Right? It is usual. Or else you will, as coming, you will not take the full suck up. As little as, whether it is a tolerable or not. Okay. Then you will start, as the timing is there. Okay, it's just 9 o'clock. We will need to go at 9:30. Let's have a lesser 10, 15 minutes, have a chatter, better talk with one to another, then we may go. It's maybe a usual, or else if you are in hurry, you start to blow. And then you may start. Same like this. Here, if you are missing some of the step, we may miss the problem. So this method, people are altering that rather to mix here. Okay, mix it here. First step before itself, so it's more easier and convenient to mix properly. And thus pour. So this method is been used in microbial growth. So earlier we have seen just what pour plate technique. Now it is being considered in application form. So this question, yeah, that is like a direct measurement. 4 to 5 method is there. All question can be asked in the 8 marks for a direct. All questions can be asked into 6 to 8 marks for the indirect method. Individual can be asked into 3 to 4. So same thing may be repeated. Sometimes it's been written here or explain the pour plate. Again, it is written as a indirect method or the direct method. So it is sometimes gifted or sometimes problematic that you will not knowing anything. And the same question is in different format is asked again. Sometimes may happens, depends upon the examiner, they may ask. So it is the somewhat more or less repetition. So one is a pour plate, one is a spread plate. Spread plate also, you may have some water sample, or sometimes I just last time I had told regarding the effect of certain metals or the chemicals. So over there, what we can do? Prepare the plates, pour the culture. You have done the spreading, just incubate. So growth will be there. Here you can put some metals, either metals or chemical. If it is a liquid, you may go for while mixing, mix with some chemical. Then also there will be a no growth. Same thing you can do in a tube. Either you prepare the plates or in a tube. Maybe it's a 10 or 20 ml. You have taken the nutrient broth. You have inoculated. Along with it, you may add certain chemicals. So when you are coming on the next day, you will observe by just naked eye also we can see, is there any microbial growth is there or not? Or else here also, if it is there, sometimes what happens that chemical either support or inhibit the growth. Or sometimes there is a no effect. It may happen. It is something is coming when you start to talk with your one or another friend, so new person is coming, right? Either you get benefited, sometimes you get harm, or there is a no any effect. The person is coming, whenever it's going, nobody has noticed. Some person is coming, okay, it is something that always problematic in a class. Everyone is noticing that, okay, something is going on, class is getting disturbed. But sometimes some person is coming that having some discussion, sometimes healthy discussion, it may be useful for to you and the other also. So always there is a three kind of effects could be possible. So that measurement we are going to do based on these points. So same thing in a different picturization form is shown into one to another. There it is like a pour plate to split plate. What we are doing in the pour plate, what we are doing into split plate. And here after you can measure or count the colonies. You may go for the colony counter, or by just using your pencil or marker, you can just at least count. If it is the more colonies, you may prepare the sectors. Sector number 1, 2, 3, 4, 5, 6. You may prepare the sectors, and according you may going to write in your notebooks. So it's both the way it is possible. Sometimes less colonies there, you can just count with normal. Or if you are using the colony counter, okay. So colony counter is having their own way to just it is like a screenshot. It's going to take the pictures, or else if it is not a computer aided, then what you can do, there is a screen like this on the place. So it is going to, you may touch the area. One knob like structure is there like a pen. So it is going to remember in their area upon the screen that something is there. When you touch this area, it's going to get counted. So different kind of colony counters are available for this experimental part. But here is some disadvantage. What could be the disadvantage of the pour plate and the spread plate? I think you have learned already. Maybe someone has written in your exams. In our second mid-semester also. Yes, sir. Huh? Yeah. તો અમારે ક્યાં બેસવાનું તમને કીધું નથી. નહીં તો કરવાનું શું હોય મને હું તો I'm new faculty here. What you will suggest I may do. If you say I will allow them to free. I'm from microbiology. I don't know anything here. What could be the formalities are there? No mail came. So what should I do? I should allow them to go in where? You may just confirm, sir. Because here the HOD has from my end is not came. So I am not able to ask to anyone. So examine unit has done any practice, or it is just
have you been informed
nothing is reinformed. That is why otherwise why should I came here? Yeah. Why not? You may just confirm. Okay. Wait a minute, best of. Just wait for 2 minutes. હું તો અત્યારે જસ્ટ જોવો છો મને તો અહીં કઈ ખબર નથી સર આ તો આજે ખબર પડી કે હોય
I would request you કે એકવાર તમે તમારા હેડ સાથે કન્ફર્મ કરી દો.
એ જ કહું છું ને અત્યારે જે આવ્યા જ ન એમાં.
Confirm it immediately because I have to start at 9. I think you will have to make it. I don't have any other.
Guys, anybody has informed you people that some exams are there in class? Nothing, but not sure. That sir is requesting that they are going to take the class. So you people just move out.
ખાલી એકવાર છે ને હેડની સાથે કન્ફર્મ કરી લો કારણ કે અહીંયા તો એક્ઝામનો પહેલેથી સ્કેડ્યુલ જ છે.
એટલે હું જ નવો જ છું એટલે તો મને કઈ ખબર નથી સર. આઈ ડોન્ટ માઇન્ડ ટુ આસ્ક.
ખાલી એમને પૂછી લો કે કોઈ બીજો ક્લાસ અગર ખાલી હોય ને તો ક્યાં કરજો કેમ કે
અત્યારે હું છે ને સ્ટુડન્ટ્સ ને કહીને ક્લાસ ખાલી કરાવી દઉં બરોબર પછી હું બારે ઉભા રહીએ ઘડીક વાર મેડ સાથે વાત કર બીજું એ રસ્તામાં એ છે બાજુ તો છે તો પ્લીઝ મૂવ આઉટ. બારે લોબીમાં ઉભા રહ્યો હું સાહેબ શ્રીને ફોન કરીને પૂછી લો કે આપણે શું કરવું એમ મને કઈ ખબર હવે જો હું મારું કામ કરું તમે તમારું કરો.